Greetings, I'm Chatya from 2nd MSc Microbiology. In this video, let's discuss about quantification of soil microflora. Soil microflora. Soil microorganisms are numerous that constitute the soil. Soil microflora are broadly defined as a group of microscopic life forms that include bacteria, archaea, actinobacteria, and fungi. Soil microflora helps decompose organic matter, increase soil fertility, promote plant growth, etc. Quantification of soil microflora. A number of microbiological parameters have been suggested as indicators of soil quality, namely microbial diversity, nutrient cycling, microbial biomass, and key species. Microbial quantification methods are divided into three broad groups, which are microbial biodiversity, microbial biomass, and key microbial species. Microbial biodiversity. Microbial biodiversity can be defined as the number of species present in a community. More traditional methods of measuring soil microbial community structure include phospholipid fatty acid analysis and substrate utilization patterns using biolog, PCR based method, that is terminal restriction fragment length polymorphism, is used to examine different populations present in a community. Biolog. Biolog is used to determine how soil microbial communities respond to specific cropping and management practices. Biolog is not recommended method because it is only applicable to bacteria and has a high degree of bias towards fast growing bacteria. This assay is also dependent on growth of cells under specific conditions. PLFA analysis. Phospholipid fatty acids are stable compound of cell wall of many microorganisms. They are specific for subgroups of microbes. Phospholipid fatty acids are extracted from soil samples and are analyzed by gas chromatography. This technique gives estimates of microbial community composition, biomass, and fungal bacterial ratio in the soil. Terminal restriction fragment length polymorphism TRFLP examines different populations present in a community. The TRFLP method involves the amplification of 16S rRNA gene with specific primers. The primers are labeled with a fluorescent tag resulting in labeled PCR products, and cut with restriction enzymes results in labeled fragments. PCR products are separated through electrophoresis according to their size, forming bands. Microbial biomass. Soil microorganisms are usually heterotrophic and they rely on carbon energy from sources outside the microbial community. Soil microbial biomass contributes to soil structure and soil stabilization, also recommended as an indicator of soil organic carbon. Determination of soil microbial biomass can be done by two methods, which are direct methods and indirect method. Direct methods, which are microscopy and plating in culture medium and counting, and indirect method, that is chloroform fumigation. Microscopy. Direct counting of soil microorganisms can be done using microscopy and is the oldest method of determining microbial biomass. The main disadvantages of microscopy are that it does not allow counting of a specific microbial species, and some stains won't differentiate between living and dead organisms. Accuracy of counts is very low and are error large. Culture methods and plate counts. Culturable populations of bacteria, either total populations or specific groups, have been used in a number of studies for determining soil health. Fungal bacterial ratio. Grasslands and agricultural soils have bacterial dominated biomass, whereas forests have fungal dominated biomass. Highly productive agricultural soils have ratios of fungal to bacterial biomass near 1:1. Soil respiration. Soil respiration can be determined either by carbon dioxide production or oxygen consumption. High respiration rates are indicators of healthy soil. Soil respiration remains the best index of whole metabolic activity of whole soil populations. It has been a traditional method since the initiation of soil microbiology. Chloroform fumigation. Chloroform fumigation is the commonly used indirect method to determine soil respiration and microbial biomass. Methods used are fumigation incubation (CFI), fumigation extraction (CFE), and substrate induced respiration (SIR). CFE measures the total microbial biomass while CFI measures the total microbial biomass under optimum environmental conditions. SIR measures the metabolically active portion of microbial biomass by measuring the initial change in soil respiration rate as a result of adding an easily decomposable substrate. Key microbial species. Microbial key species in soil can be defined as organisms that fulfill important functions in the soil ecosystem. For example, mycorrhiza. Mycorrhiza. The abundance and diversity of mycorrhiza is determined by the extraction of spores from soil samples and subsequent counting under a microscope. Methods for direct detection and quantification of mycorrhiza in soil samples include 18S rRNA and nested PCR. Suppressive soil. A suppressive soil is a soil where disease suppression occurs. Antagonistic microorganisms that produce antibiotics usually dominate the microflora of disease suppressive soils. These include Penicillium, Trichoderma, Aspergillus, Streptomyces, Pseudomonas, Bacillus, Rhizobium, Acetobacter, Azospirillum, etc. The presence of plant pathogens in the soil may indicate the existence of soil health problems such as nutritional imbalance. Monoclonal antibodies, DNA probes and PCR can be used to detect beneficial as well as detrimental species that are associated with healthy as well as unhealthy soil. Molecular probes, FISH and dot blot. The molecular probe approach includes fluorescence in situ hybridization and dot blot. These techniques are based on the design of labeled oligosaccharides that specially target the 16S rRNA of specific organisms. FISH is performed on fixed whole cells with fluorescently labeled oligosaccharides and are observed by epifluorescence microscopy, while in dot blot, nucleic acids of cells are hybridized with radioactive labeled probes. Thank you for watching.